Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This one's the most important one in the genetics inheritance playlist. This is genetic inheritance itself. We're looking at genetic crosses now and the mechanism by which genes are passed on from one generation to the next. So the previous lessons on the structure of DNA, protein synthesis, meiosis, asexual and asexual reproduction have all been building up to this one. They gave you the really solid background to now be able to access this genetic inheritance lesson. But what I've done for the first couple of slides of this lesson is I'm going to give you some background information. Now, this has taken me years to work out as a teacher what are the key things that you must know before you can understand the genetic crosses themselves. So this video has a lot of information. The slides aren't as bright, as colourful as most of my presentations. The first few are just chunks of really vital information, but they are essential. So take plenty of time on them, really think about all the points that I'm giving you. This is from the GCSE specification. Obviously at A level, there's a huge amount more that crops up in genetic inheritance, but this video is just suitable for the GCSE syllabus. As always, please do subscribe to my channel if you haven't done so already. Thank you. So this is the essential information. I cannot stress strongly enough. Please do not move on unless you fully understand all of these points. Because if you haven't got this background information already in your head, you're going to get totally lost by the next few slides. And if there's things that crop up on this slide that you think, oh, I don't really get that, I'll try and direct you back to the videos that can fill that gap in. Okay, this is the most difficult part of the GCSE course, I think. Some people just really struggle to get their heads around it, but I'm going to make it as simple as I can for you. So point number one, DNA is the coded instructions for making polypeptides which fold into proteins. If you don't know what I'm talking about, go back to the video on detail of protein synthesis. It's 3 presentations ago in this playlist on genetic inheritance. So DNA gives you the code, makes that copy that can go out of the nucleus, goes to a ribosome. Ribosome reads the bases on the nucleotides 3 at a time to get the amino acids to stick together in that polypeptide chain. That folds up into the functional protein. That's really essential information. 2, proteins do everything. I've said it so many times in my presentations, but really keep it in mind. Proteins do everything. Therefore, the DNA, the sections of DNA coding for those proteins are vitally important. A gene is that section of DNA that codes for one polypeptide that will then fold into a protein, of course. But, and this is new information, there's more than one version of any one gene. Okay? What that means is that the different versions produce slightly different versions of the protein that does the same job but in a slightly different way. So for example, if you came to my house and I said, oh, can I get you a drink? You said, yeah, lovely, can I have a glass of water? I could bring it to you in a glass or a cup or a mug or a beaker or a jug or anything. It doesn't matter. They're all carriers for the liquid that's going to go to you. They're all different versions of just a carrier for some liquid, but they all do the same job. That's really what we're talking about with different versions of genes. It's a protein that has the same basic function, but it can do it in slightly different ways. That's really important, that there are different versions of genes. Now, the different versions of genes are called alleles. That's a really important keyword that we're going to be using again and again in this presentation. So an allele is a different version of a gene. Now, you inherited a full set of genes from each parent. Remember back from the meiosis lesson, you got 23 chromosomes from your mum, 23 from your dad, and each set of 23 carry a full set of human genes. So you could make a human from those 23 chromosomes. Okay, so you get a full set of genes from each parent. It's 23 from each, giving you 46 in total. Now, what that means, and this I've worked out is the crucial thing to understand with genetics, you must remember that that means you have 2 copies of every single gene. So the genes that control your eye color, you got one from your mum, you got one from your dad. Gene that control whether you can make insulin, you got one from your mum, you got one from your dad. There's 2 of everything. That's really important. They can be the same allele or different alleles. So you might have got for your eyes an allele from your mum that says have brown eyes. From your dad, you might have got an allele that says have blue eyes. It doesn't matter. You've got a gene that controls color protein that's in your eye, but you've got 2 versions of that gene. They can be the same or they can be different. When a gene gets read and its protein gets made, we say it's been expressed. Expressed is a vital word when it comes to genetics. So expressed literally means the bit of DNA got turned into a protein. It expressed itself, just as I'm expressing myself now by giving words. A piece of DNA gives the protein to become expressed, so its function gets expressed. Some alleles are more easy to express than others. This is vital. Alleles that are easily expressed, as in alleles that can easily be read by the body, made into the little copy that goes to the ribosomes. Ribosome can easily turn them into protein. Alleles that are easily expressed are said to be dominant alleles. Alleles that are a bit harder to express are called recessive alleles. Now, your cell only needs to express one allele for any gene. I only need to follow my mum's one or my dad's one to have the proteins that perform the function I need. But this is the vital bit. Your cells will always take the easy option when it comes to the two copies. I'm going to try and make this a bit more clear by putting a little diagram above my head as if the gene was a form of greeting and the alleles are different forms of a greeting. Okay, so if I put up first the two versions of a greeting that I got, I put up hello and hello, two that are the same. They're alleles of a type of greeting, if you like. So I can have hello and hello. They're both easy to read. They're both the same. So I'll just say hello. Okay. Now, if I've got two that are different and one is dominant and one is recessive, so if I pull up hello and then smaller and in difficult to read script, good morning, I only have to read one of those. And if someone says, oh, you've got to give a greeting to this person, here's your options, and I've got those two, I'm going to glance at it and go, hello, because that's far easier to read and express than the good morning that I've got written there that's quite difficult. However, if the two versions I've got are both that difficult to read good morning, and someone says, you've got to say one of these, and suddenly I'd have to go, good morning, I've got to read one of them. They're both equally difficult to read, so you just pick either of them. Okay, hopefully that makes sense. If any of these points haven't made sense to you, go back and have a look at them, rewind the video, have another look at them again. But hopefully they're straightforward, but they are essential to the next understanding. Plus you've got a bunch of new keywords there. Allele, dominant, recessive, expressed. Make sure you know them. Take a screenshot of this for your notes and then we'll move on. Okay, we're going to look at something called genotypes and phenotypes now. So two more keywords. There's lots of keywords in genetics, but I'll break them all down again. So genotype is the combination of alleles you have for any one gene. I'm going to make that more clear because remember, you've always got two. Now, if you've got both your alleles the same and they're the dominant version, then your genotype is said to be homozygous. Homo, because it's from the Latin homeo, meaning the same. Zygous meaning genes or information. So homozygous meaning same gene, same allele. And because they're both dominant, it's homozygous dominant. So if you're two alleles, you've always got two copies, remember, and if they're both the same and they're both the dominant allele, you're homozygous dominant. That, using my diagram from before, is the hello, hello, the two dominant greetings. If you're hello, hello, you're homozygous dominant. Both alleles are the same, they're both the dominant version. If they're different, one dominant and one recessive, it's heterozygous. Hetero meaning different. That's the equivalent of the hello, good morning. Okay, one was really obvious, one was more difficult. They're the different ones, one dominant, one recessive. If they're the same but they're recessive, hopefully you work this one out. It's homozygous, but this time it's homozygous recessive, and that's the good morning, good morning. Okay, again, key terms you must know. Homozygous dominant, heterozygous, homozygous recessive. Those are the three genotypes which show the two alleles that you've got, the combination of alleles. This is getting tricky already, I know. You may have to rewatch that to get it set in your head, but I'm trying to make it as straightforward as I can. Let's move on to the phenotype, because the phenotype is the resulting protein or characteristic that actually gets expressed. So if we're talking about my examples up there, it's given those genotypes, what do I actually end up saying? Well, for the top one that was hello, hello, I express hello. So the phenotype, what I do is hello. For the middle one, where it's hello, good morning, I express hello because I ignore the good morning, and so on. So let's look at those. For the three genotypes over there, you can see the homozygous dominant, I can express either allele, the hello or the hello. So I show the dominant phenotype, the hello phenotype. The heterozygous, dominant allele gets read because it's easiest. The recessive gets ignored because it's difficult. So again, I show the dominant phenotype. So if you were just meeting me and I said hello, you wouldn't know if I'd read hello, hello, or hello, good morning, because the result is the same. So whether you're homozygous dominant or heterozygous, you still do the dominant thing, the dominant phenotype. It's only if you're homozygous recessive and you've only got the option of the recessive that you show the recessive phenotype. So it's only in the circumstances of me having good morning, that I would actually say good morning. Hopefully that makes sense. But that's really, really important about how you express a genotype into a phenotype. And remember, you have 2 copies of every gene, 1 from each of your parents, but meiosis splits them apart when making gametes. So only 1 gets passed to your offspring. So if I had the heterozygous one up there, the hello, good morning, that's in my cells. When I make my gametes through meiosis, I separate them. So half my gametes would have the hello, half would have the good morning. And that's how it works when you're passing it on to your offspring. Right, that's the end of the 2 essential background slides. We're going to move on to the genetic crosses now. But if there's anything in these first 2 slides you haven't understood, go back and have another look through them. And take a screenshot of this one to add to your notes as well. Right, hopefully everything has made sense to you from the previous slides, because we're now going to put that into practice with an actual genetic cross. Now, we're not going to look at a human example. I'm going to jump across and we're going to look at fur colour in gerbils. So if I've got these 2 gerbils next to me here, I've got the lighter one and the darker one. But what we're talking about with their fur colour is that it must be controlled by a fur colour gene. There must be a gene, a bit of DNA, that codes for the colour of their fur. And that gene must code for a coloured protein that's in the fur. Now let's say, we've got 2 mice here, so let's say there are 2 alleles that control the fur colour. We've got a black allele, giving the darker mice on the far side of me, and we've got a brown allele, giving the paler brown mice closer to me. Now let's say the black allele is dominant, the brown allele is recessive. For the black dominant allele, we're going to use a symbol of a capital B to represent it, because when you're doing crosses, to save you writing everything out, you use symbols. So you're going to use a capital B for the dominant one, and we're going to use a lowercase b for the recessive one. Now, by convention in genetics, you use the same letter, and you use an uppercase version of it to show dominant and a lowercase version of it to show recessive. Now, that's easy when you're using something like a B. Please, if you're writing your own, avoid using letters like O, because O is just a circle. It's very hard to show a big or a little, an upper or a lowercase O. It's not a good letter to use. L, quite similar. Some people don't do their L's very neatly, or they put a curve on the bottom. L's can be easily confused. You know, pick letters that are really different, like an R or a Q or a B or a D or any of those ones, okay? But we're going to use B here to show the dominant and recessive. Now, for the genotypes, remember we've said there's only 3, homozygous dominant, heterozygous, or homozygous recessive. We're using our code, the B's. The homozygous dominant will be big B, the heterozygote will be big B, little b, one of each, and the homozygous recessive will be little b, little b. If we think about the phenotypes that those gerbils would show, the homozygous dominant is going to be black, have black fur. The heterozygote, because it reads the dominant one and ignores the recessive one, because the recessive one is difficult, that's also going to have black fur. And the homozygous recessive, because it's only got a brown allele, is going to be brown. So the dark mouse over there, the black coloured mouse, you don't know by looking at it whether it's a homozygous dominant or a heterozygous gerbil, because both of them would lead to that black coloration. Okay, so let's put that information now into a cross above my head. Okay, so let's look at an example of a genetic cross. Now I'm going to show you a way to set out your genetic crosses that are absolutely foolproof to guaranteeing you the marks in the exam, but you must do certain things. So first off, in the exam, whatever your information you're given, I want you to write down parent 1 and parent 2 so you know what you're talking about. Then I want you to write these 6 things down. Okay, or you don't have to write these 6 things down in the exam, but when you're practicing, try and get used to writing them down so that you keep these 6 things in your head, because these are what I always want you to show. the parent phenotype, so what the parents look like, the parent genotypes, what alleles they've got, what gametes the parents would make, what their offspring genotypes would be when you rearrange the gametes, what their offspring phenotype would be based on their genotype, and then what the ratio is, how many of each phenotype do you get. If you can hit those 6 things in any of your exam questions, you're going to be guaranteed the full marks. So let's put some information in here. Let's say I'm crossing a black gerbil with a black gerbil, okay? But if I give you the genotypes and I say that both of them are heterozygous, so they're both big B, little b, okay? So in all of their body cells, their somatic cells, they've got the big B, black allele, and the little b, brown allele. Now, by the process of meiosis, if you think those 2 different alleles, those are on the maternal and paternal chromosomes in a homologous pair. So those get separated during meiosis 1. So half of the gametes are going to end up having the big B, half of them are going to end up having the little b. So all you need to show on the genetic diagram are what splits from that the parent genotype, okay? So for that parent 1 that was black phenotype, big B, little b genotype, his gametes are going to be half big B, half little b. Other parent, because he's got exactly the same genotype, is going to be exactly the same. Now, something I always really pester my students to do is put circles around gametes. The only reason I really pester them to do that is because when you're writing them quickly in the exam, if you're in a rush, if you don't leave a big enough gap between them, then it looks exactly the same as the genotypes. It's a big B and a little b. All you've done in the next line is put a gap between them. Drawing a circle around them shows that you've recognized that they've separated from each other at meiosis and now they're individual genes. Remember, gametes only contain half the information, 1 set of DNA, 1 copy of each gene. So please get used to using the circles. Right. Try and follow the cross that I'm about to do now, because I'm going to do it in quite an old-fashioned way, the way that I learned it. Then I'll show you a quicker way that you can use, okay? But what you've got to imagine, let's say parent 1 is the male. That means those gametes are sperm. So that first sperm carrying the big B could fertilize an egg with the big B. You get those 2 linking together. One parent gave a black allele, the other parent gave a black allele. The genotype is big B, big B. But of course, sperm with the big B could just as easily have fertilized the other egg with the little b. So it could have got that result, forming a heterozygote. Now, there's no other options for the sperm from that from parent 1. So you move to the second one, the little b, which could have fertilized the first, giving another big B, little b. And just like when you're writing a name, the capital, the dominant one always comes first. That's why you might have thought I've got them the wrong way around. And lastly, the little b, the brown allele, could go with the other little b, the brown allele from the other one, giving you little b, little b. Now, the quicker among you may already have worked out what the phenotypes will be. The big B, big B, the black is going to give you black. The black brown heterozygote is going to give you black, as is the next one. It's only this last one that's the homozygous recessive that's going to give you a brown phenotype in the offspring. So your ratio of offspring is 3 to 1, okay, 1 brown. Now, this box contains all of the information that you would need to show for a genetic cross in the exam. You really, at this stage, just need to understand the mechanism behind what I've done up there to show how things can pass on. And it's really useful if you understand that 2 black mice can still have a brown offspring because they both carry the brown allele. And in the only 1 out of 4 chance that they both pass their brown allele on, you'll get a brown mouse. Okay, really important that you've understood that. So take a screenshot of this, but then I'm going to show you an easier way of doing that quite messy cross that's above me. So take a screenshot of this, and then I'm going to clear some space. Okay, hopefully you've done that and it's ready to add to your notes. So I'm going to get rid of my messy cross, and I'm going to show you a way called using a Punnett square. This is probably what you've been shown in. So I'm going to bring up a little grid structure. Okay, there's my little grid structure, and I'm going to put parent 1's gametes in the side of the grid, still in circles, and parent 2's gametes on top of the grid, still in their circles. And then all you do is kind of come across and down, and every time you hit, you fill in what would be in the box. So the first box in the corner there, you come down from the big B, across from the big B, and the genotype's the big B, big B. Okay, you do that for all 4 and you get exactly the same as you would have done with my more messy arrows. And of course they still translate. That one's your black one, that's a black one, that's a black one, and that's your brown one. Okay, so this information in this box is perhaps a more straightforward way for you to show across. So take a screenshot of that as well, and then we're going to work through a few more examples to try and reinforce this. But if any of this didn't make sense, go back and watch it again, because these are genetic crosses. These are what you're going to be asked to do in the exam. So sticking with the same gerbil example and fur colour and looking at the black and brown alleles, let's pull this question up. What would be the result if a brown gerbil was crossed with a black gerbil? Now, if that's all you're given in the exam, you've got to do some deduction work here. Always start though with that same framework, okay? Think parent 1, parent 2, and the 6 stages that you need in a genetic cross, and start to think, right, well, what on here can I fill in from the information they've given me? Well, they've told you it's a brown and a black gerbil, so they've given you the phenotypes, so you can fill those in. Then you need to think, can I deduce the genotypes just from that information? Well, you can. The one you can fill in is the brown gerbil, because there's only one genotype that gives you a brown phenotype, and that's homozygous recessive. So you know the brown gerbil must be little b, little b. Now look at the black gerbil, and hopefully you've spotted a problem, because the black gerbil we can't be as sure about, because there's 2 possibilities of genotypes that lead to black gerbils. So what we're going to do now, we're going to pull up 2 options here, because what it could be, if we go back over to option 1, it could be that our black gerbil is black. Both copies of its genes, both alleles are black. Or in option 2 above me, it could be black brown. It could be the heterozygous genotype. Okay, let's work both through. Let's look at option 1 to start with, and let's put the names to these crosses as well. So option 1 is if it's a homozygous recessive crossed with a homozygous dominant. Option 2 is if it's a homozygous recessive crossed with a heterozygote. Okay, let's go back to option 1 though. Let's pull up our Punnett square, our grid. Let's put the gametes for parent 1. Now remember, you've just got a little b and a little b. That's what's in the normal somatic cell. They separate by meiosis, so what we can fill in are the 2 little b's. The other parent's only got 2 big b's, so when they separate at meiosis, you get the gametes both with a big B, with a black gene. Now, looking at those, the one parent's only got black alleles to give, the other parent's only got brown alleles to give. So hopefully you can see all 4 offspring will be heterozygotes, because they've got a black from one and a brown from the other. They're all going to have 2 copies, one black, one brown. Okay, which means when you look at the offspring phenotypes, if they're all heterozygous, they're all going to be black. So your ratio is 4:0. So if you cross a brown mouse that's homozygous recessive with a black mouse that's homozygous dominant, all of the offspring will be black, because every single offspring will contain 1 black allele from the one parent, the parent 2, and if they've got 1, they'll express it because it's easier to express because it's dominant, and all of the offspring will be black. Okay, compare that with option 2 up above me now. Same grid, same allocation of gametes there, but now notice for the parent 2, we've got the black and the brown alleles in the separate columns. So now when we fill in the grid, we've got a heterozygote and a homozygous recessive, and another heterozygote and another homozygous recessive. So work out their colours. The heterozygotes are both going to be black, just like in option 1. But now we've got 2 homozygous recessives that are going to be brown. So now just that one tiny change between option 1 and option 2, just changing whether parent 2 was a homozygous dominant or a heterozygote, we've changed to a ratio of 2:2. That's quite a significant difference there, depending on one parent. Now, looking back at the original question, what would be the result if a brown gerbil would be crossed with a black gerbil? We don't know. It could be either of these 2. All the offspring could be black, or half of them could be black and half of them could be brown. So they'd never actually ask you that question in the exam. They'd give you some more information, and using that information, the most important thing you can do is look at the ratios in the offspring, if it gives it to you, because that can help you deduce the genotypes of the parents. If all of the offspring were black, then you would know for a fact that the one parent had to be homozygous dominant. If it was a more of a 50-50 split, then you'd know it's this cross where they're a heterozygote. So actually in the exam, they're more likely to give you something like this. Explain how a brown and black gerbil could produce offspring where half of them are brown and half of them are black. Well, that's what we've got in this one. So your answer would literally be what's inside this red square. If you wrote down in your exam paper exactly what's inside this red square, guaranteed full marks. You've given the parent phenotypes, the parent genotype, the gametes, the offspring genotypes, the offspring phenotypes, and the ratio. This diagram fully explains how that can happen. Okay, hopefully that's made sense. Take a screenshot of this. Okay, last slide, and this is just advice, because these videos take you through the theory. They teach you the understanding. But in my classes, I would spend 3 or 4 full lessons, hour-long lessons, teaching this genetic stuff, because I'll be giving my kids loads of examples, loads of alternatives. We do different genes in different organisms, lots and lots of practice, because it's practice, practice, practice that makes you good at genetics. So my advice to you, knowing that genetics exams questions are notoriously tricky, and they're tricky because there are many ways in which they can present the information. They could just literally list it out like I did before, saying here's the gene, here's the alleles, this is dominant, this is recessive. Or they might give you a paragraph just saying, oh, there's loads of mice or whatever, and most of them are this, and there's very few that are that, from which you'd have to deduce, oh, well, that one must be dominant, that one must be recessive. They might give you a family tree diagram asking you to work out why one is one thing and one is another. They may give you just pictures to try and interpret stuff. There's loads of ways they can present it. And there are many ways to phrase the question. They could explain how this happens, or they could say, draw a diagram that shows this, or they could say, what would the parent genotypes be if this was the outcome? There's so many ways they can give you the information and phrase the questions. So the more exposure you can have to questions, the better. So what I want you to do, find past exam questions. Go to the exam board websites. They always have loads on there. Go online, search for past papers. There's loads available. Use the questions in your textbook. Go to the chapter on genetics. Generally at the back of the chapter, there will always be loads of practice questions. On each page of information in your textbook, there's normally questions to answer as well. Get hold of some other textbooks. You know, for every exam board, there's about 6 or 7 different textbooks from different publishers that all have different exams that you could practice on. Also, pair up with a friend once you get pretty good at it and set each other questions. Just make up a gene, make up the alleles, make up what's dominant and recessive, and set each other some tasks to try and work out. Or do a bit of research and find some fun things like the genetics for being able to roll your tongue into a tube, or whether you've got dangly earlobes or not, or whether your thumb goes straight up or goes weirdly backwards like mine does. It's all genetically controlled. Go and find out and set questions to each other. Really important to do as many as possible. This is the real skill on those though. Practice how to find the key facts from the information that's given. And those key facts are what alleles are there, what's dominant and what's recessive, what are the parent phenotypes, and do you know the genotypes? Because if you've got that information, you can do the whole cross. What ratio do you observe in the offspring, and what does that tell you? If they're only giving you the offspring and the ratios, you can basically work out what the parents will be, because we've covered all 3 potential crosses in this video. The first one that I showed you was a heterozygote with a heterozygote, and you've got a 3-to-1 ratio. The second one we looked at was a homozygous recessive with a homozygous recessive, and you've got 4-to-0. The other one was a homozygous recessive with a heterozygote, and you've got a 2-to-2. That's pretty much it. You're just looking for those ratios to work it out. But if you can answer those 4 questions, or pull out the information to answer those, then you'll be doing really well. Because if you can answer those, you'll use them for the 6 key levels when you build an answer, which are these things up there. As long as you can, from the information given, pull out the information to answer those 6 things, or use it to work out those 6 things, you're going to get full marks in genetics. Hopefully this hasn't been too much of a challenge and it has made sense. But the next video is going to add some clarification. So the next one is examples of genetic conditions. Now there's several conditions you need to know. It's part of your syllabus. But what I've done in this next video, I'm going to look at that condition and we're going to do a few genetic crosses or look at a few different ways in which you can present the information, like a family tree diagram. So this next one will really reinforce all the theory that I've taught you in this video. But if you're at all unsure, go and watch the video again. Use the first couple of slides to direct you back to any previous videos that you think you might have gaps in your knowledge. But hopefully this has all made sense. As always, I'll ask that you please do subscribe to my YouTube channel if you haven't done so already. Like this video if you find it useful, and head over to my Facebook page. There's loads of quite interesting random biology infographics that I've made on there that hopefully you'll really enjoy. Thank you. Good luck.